Hi class, today we're going to talk about metabolic regulation and the various layers of regulation that organisms use to control metabolic activity. So as a high level overview, there are a lot of different layers of metabolic regulation. There's, of course, these classical mechanisms that are familiar from introductory biochemistry, including allosteric regulation and covalent regulation of enzymes. There's transcriptional level control that affects the level of each of the various enzymes. There's multi-enzyme or multi-organ interactions that help regulate metabolism. And there's compartmentalization that keeps some enzymes sequestered or some pathways sequestered from others. And all this can be coordinated through hormonal control or these signaling mechanisms. So we're going to go over examples from each of these today. And we're going to talk about how they all interact to regulate your body's metabolism. So allosteric regulation is one of the most classically discussed metabolic regulatory mechanisms available inside the body. And these are acting typically in these sort of short-term time frames. Responding to conditions as they change in sort of relatively short time scales inside the body. The way they work is an allosteric activator and inhibitor will bind to an enzyme, change its conformation, and that change in conformation will ultimately result in an altered enzyme activity. So there are two main categories of allosteric regulation. There's feedback inhibition and feed-forward stimulation. So in the case of feedback inhibition, the products at the end of the pathway can inhibit something upstream. Typically, this will be an enzymatic step that involves a commitment of energy, or is otherwise a committed step for one reason or another. There's also feed-forward stimulation in cases where you have an intermediate that stimulates the downstream catabolism of a metabolite. You see this when it's needed to prevent the buildup of some particular molecule. So we'll talk about examples of each of these in the context of glycolysis and the TCA cycle today. Now, for glycolysis and other energy-related pathways, the overall energy state of the cell is a major regulator of metabolic activity. So AMP ATP ratios have a direct impact on the activity of the rate controlling steps in glycolysis. So these steps, the committed steps in glycolysis are hexokinase, PFK, and pyruvate kinase. Now, each of these steps involves either the commitment of the first phosphate onto glucose, the commitment of the second phosphate onto glucose, or the ultimate production of pyruvate and ATP. So these are the three regulatory points in glycolysis, and they're regulated by two different mechanisms. Glucose 6-phosphate actually feeds back and inhibits hexokinase itself, and phosphofructokinase and pyruvate kinase are each inhibited by ATP, which is sort of the end product of the pathway. And on the reverse side, for when the energy state is a low and AMP levels are high, phosphofructose kinase is stimulated, and so is pyruvate kinase, and that leads to higher glycolytic rates. Now, these are not the only allosteric modulators of metabolism. In the context of glycolysis, there are quite a number of different molecules that affect the activities of these rate controlling steps. So AMP, as we just talked about, AMP, ATP, but also citrate and protons. Citrate is a product from the TCA cycle. So this is sort of reaching out from the feedback inhibition from a far downstream pathway that can then inhibit PFK way upstream, and protons, which would be produced in the case of anaerobic metabolism. Similar with the pyruvate kinase, you have ATP inhibiting pyruvate kinase, but also alanine, which is just one transamination away from pyruvate. So it's very similar to feedback inhibition from pyruvate. There's also regulation of the two steps in gluconeogenesis that bring you from pyruvate back to phosphoenolpyruvate. Pyruvate carboxylase is inhibited by ADP, but stimulated by acetyl-CoA. So when you have lots of acetyl-CoA, it tends to stimulate gluconeogenesis. And then up here at the fructose 1,6-bisphosphatase, it's stimulated by citrate. Again, lots of product in the TCA cycle stimulates the conversion of TCA cycles and into glycolysis via gluconeogenesis. Now, I've left out one of these really classic modulators here, and we'll talk about it more in detail. It's fructose 2,6-bisphosphate. This one is a really potent activator of PFK, and we're going to talk about how that works. So fructose 2,6-bisphosphate, it looks a lot like the glycolytic intermediate, except the phosphate is on position 2 instead of on position 1. Then happens to is it binds to and has a separate regulatory binding pocket in phosphofructokinase PFK, and that stimulates it and then stimulates glycolysis. So fructose 2,6-bisphosphate is made by this bifunctional enzyme that both can catabolize the synthesis of fructose 2,6-bisphosphate from fructose 6-phosphate, and also can catabolize its breakdown. So it's, can both make it and break it down. And we'll learn about how that bifunctional enzyme is turned on and turned off. But first, we have to talk about a covalent regulatory mechanism, because that's the mechanism through which it acts. So one of the ways that we coordinate physiology between different organs in your body, and to coordinate sort of longer term physiological shifts inside the body is through the change in phosphorylation state of the different enzymes. And so we talked about this extensively in the case of glycogen phosphorylase, where we talked about the dephosphorylated and phosphorylated states shifting the equilibrium between the active and inactive forms of glycogen phosphorylase. But this mechanism extends to a lot of other different enzymes. As a reminder, the way this signaling mechanism works is that glycogen or epinephrine will bind to a receptor here on the cell membrane. It ultimately triggers a cyclic AMP signaling cascade, which results in the activation of protein kinase A. This is a protein that goes and puts phosphates on other proteins. So there is a reciprocal and antagonistic pathway here that signals through the insulin receptor, and the insulin receptor ultimately triggers the stimulation of this protein phosphatase A, or PP1. This one removes many of the phosphates that are put on by protein kinase A, and we'll talk about how these antagonistic activities of protein kinase A and protein phosphatase 1 ultimately affect the activity of many important regulatory steps in glycolysis and other central carbon intermediates. So this really potent activator, fructose 2,6-bisphosphate that we're just talking about, that is synthesized by this bifunctional enzyme. As it turns out, the activity and the synthesis of this important potent activator of glycolysis is ultimately regulated through this reciprocal kinase phosphatase interaction we talked about. Now, protein kinase A will put a phosphate onto this PFK2, which is an enzyme that synthesizes phosphofructokinase, 2,6-bisphosphate rather. When protein kinase A puts the phosphate on PFK2, and it inhibits it, and then ultimately that results in the inhibition of glycolysis. And in reverse, when you have the stimulation of protein phosphatase, that removes a phosphate from this PFK, that enables the phosphatase activity of the enzyme to occur. And that breaks down fructose 2,6-bisphosphate to fructose 6-phosphate, and then ultimately leads to the stimulation of glycolysis. Okay, so here's a case where the production and degradation of this important allosteric activator, fructose 2,6-bisphosphate, is regulated through the antagonistic kinase and phosphatase activities that are triggered through these two signaling cascades that I just discussed. Those aren't the only targets, though, in glycolysis. Protein phosphatase A and protein kinase A act on many other targets in central carbon metabolism. Another example is their activity on pyruvate kinase, which, as you know, is the step that ultimately produces pyruvate and ATP at the end of glycolysis. So in a very similar mechanism, the protein kinase A puts a phosphate onto and then deactivates pyruvate kinase, and a protein phosphatase removes the phosphate and activates the pyruvate kinase. So in a very similar mechanism here, these kinase and phosphatase ultimately control the activity of this pyruvate kinase. So these reactions also affect the bridge cycle activity and the gateway end of carbon into the TCA cycle. Now, this step is actually regulated through a number of different molecules: ATP, acetyl-CoA, NADH, and ADP, all affecting the activity of this transition between pyruvate and acetyl-CoA. The way this works is actually happens through an intermediate. It's this enzyme called pyruvate dehydrogenase kinase. So this is an enzyme that puts a phosphate onto pyruvate dehydrogenase. Okay. Pyruvate dehydrogenase has two forms, an active and inactive form. It's inactive when it has a phosphate on it, and it is active when it is dephosphorylated. So pyruvate dehydrogenase kinase actually puts that phosphate on and inactivates the enzyme, whereas pyruvate dehydrogenase phosphatase takes it back off. Now, the activity of pyruvate dehydrogenase kinase, this regulatory enzyme, is altered by the presence of NADH and acetyl-CoA, where NADH stimulates pyruvate dehydrogenase kinase, and acetyl-CoA stimulates it as well. And in both cases, this makes sense. This is a product of the TCA cycle, and this is a product of the bridge reaction too. This is a product of the bridge reaction. So the net effect is that NADH and acetyl-CoA each act as a sort of feedback inhibition mechanism that's regulated through this independent kinase. So there's also a feed-forward stimulation mechanism that happens with pyruvate that also is mediated through this pyruvate dehydrogenase kinase, where pyruvate binds to and inhibits pyruvate dehydrogenase kinase and enables the phosphatase activity to occur, ultimately stimulating the bridge cycle activity. ADP also acts as a positive activator of pyruvate dehydrogenase. So how are all these things signaled? So in order to coordinate these covariant modifications, you have to have signaling between the different organs. This is enabled through a variety of both peptide, steroid, and other small molecule hormones that ultimately work together to coordinate the flow of energy around the body. We're going to talk through a few of these examples and talk about how the coordination of multiple enzymes in the pathway, multiple organs in the body is ultimately coordinated through these hormones. So the mechanisms that we talked about already in this class include glucagon and insulin signaling, where glucagon and insulin are both produced by the pancreas, and glucagon is secreted by the alpha cells, insulin is secreted by the beta cells in the pancreas. That's secreted to the bloodstream, ultimately acts on the liver here to either bring in glucose or turn it into glycogen, or to secrete glucose and to feed the rest of the body. There are also peptide hormones, though, that regulate this whole body energy metabolism. Leptin is a good example. This is a satiety hormone. It's one that signals that you're full. There's also ghrelin, which is a hormone that's secreted by your stomach and tells your body to eat. So all of these guys are examples of peptide hormones that ultimately trigger metabolic actions inside the body at a sort of a larger multi-organ level. Signals can also be transmitted by mechanisms that act onto the protein phosphates and kinases that we've talked about previously. One example is epinephrine or norepinephrine. They're very similar molecules. It's just norepinephrine is secreted by the nerves and is used as a neurotransmitter, and epinephrine is secreted from the adrenal glands, is used to affect metabolic activities in the fight or flight response. Both of them are synthesized from tyrosine, and they are good examples of modified amino acid derivatives. And we'll learn more about how these amino acids are synthesized in later lectures. Both of these guys are the epinephrine and glucagon, and have their effect, and the signal is transferred into metabolic action through a signaling cascade. In this particular case, it's mediated through a cyclic AMP mediated cascade, where you take ATP, you cyclize it into this potent signaling molecule here, and that ultimately leads to a cascade of phosphorylation that leads to the activation of protein kinase A, and ultimately the activation of glycogen phosphorylase, which leads to the breakdown of glycogen. But that isn't the only reaction. I know we've learned about that one extensively in this class, but this signaling cascade has a number of effects inside the cell. It can stimulate glycogenolysis. It can encourage the translocation of the glucose 4 transporter to the cell membrane. This is very important in muscles. It can inhibit glycolytic enzymes directly, and it can lead to long-term increases in mitochondrial biogenesis. And these signaling mechanisms have a lot of effects throughout the body. So some of the longest term actions in metabolism occur through transcription. So if you have a major shift in your diet, for example, from say a carbohydrate-based diet to a fat and protein-based diet, your body will adapt to it over time, and those adaptations occur through transcriptional mechanisms. There are two major categories of transcriptional regulators. They're activators and repressors. An activator will bind to and turn on the transcription of a silent gene, whereas a repressor binds to and turns off the expression of a target gene. One of the most famous examples of this is the lac operon. This mechanism is used in prokaryotes to regulate the expression of beta-galactosidase and other lactose catabolizing related proteins. Briefly, the way this works is that there's a repressor, LAC-I, that binds to a regulatory element upstream of the operon. The LAC-I, binds to this point and then prevents the transcription of this operon. However, when allolactose, which is a side product of the beta-galactosidase reaction, when it binds to this enzyme, it releases that repression and allows the entire operon to be transcribed, which includes the production of the enzyme, beta-galactosidase, this permease enzyme, and acetyl transferase enzyme. Now, this entire operon here has a small amount of leaky expression, which is how you have sufficient both lactose permeability and beta-galactosidase activity to turn on and turn off the repression of this LAC-I. But this mechanism is a great example of how the need for a metabolic enzyme is ultimately regulated through a transcriptional mechanism. So that's a prokaryotic example. In eukaryotes, especially in vertebrates, there are a lot of other transcriptional regulations that are transcriptional regulatory mechanisms that are at work. So insulin that we've been learning about, this insulin signaling pathway, is a good example. It triggers both a signaling cascade that leads to the production of MAP kinase. So MAP kinase is a long-term regulator of cell growth that is very active in many kinds of cancer cells. Also has lots of effects on gene expression and protein synthesis and glucose transport. It has a number of other insulin has a number of other effects. Many of these also have interrelationships with oncology. For example, FOXO and TOR are good examples of that. But in addition to these more familiar signaling pathways, there are ones you probably haven't heard about. The steroid response element binding protein is a great example. It can be triggered through PI3 kinase from insulin signaling, but it can also be triggered by sterols directly. And this one has a direct effect on metabolic pathways inside of glycolysis, including targets in fatty acid synthesis and other places. We'll learn more extensively about this during our lipid section. There's another example is the carbohydrate response element binding protein. This one's really cool, and I'm going to tell you a little bit more about it on the next slide. So the carbohydrate response element binding protein is unusual in that it binds a metabolic intermediate, xylulose 5-phosphate, which is an intermediate of the pentose phosphate pathway. When it binds to this protein, CHRBP, transfers to the nucleus, turns on the transcription of quite a number of other downstream metabolic pathways, including enzymes, including pyruvate kinase, malic enzyme, ATP citrate lyase, acetyl-CoA carboxylase, fatty acid synthase, and steryl-CoA desaturase. So it turns on lipogenesis and fat storage. The cool thing about this is all regulated here through this, signaling of the pentose phosphate pathway. It's a pretty unusual mechanism as a potent regulator of lipid biosynthesis through glycolysis or glycolytic intermediate. So there are other potent regulators of this long-term transcriptional regulation. That includes steroid hormones. So steroid hormones, we're familiar with in the context of androgens and estrogens, these sex hormones that have an impact on everything from menstruation, lactation, and anabolism, and a variety of other things. But the steroid hormones are a sort of wider class of hormones. They're all powerful signaling transcriptional activators, and they have effects really wide-ranging throughout the body. So as a brief overview, a couple of examples of this include the secretion of cortisol, which is a stress hormone from the adrenal cortex, which has effects on whole body metabolism and affects a wide variety of functions throughout the body. There are effects of a variety of hormones on the kidney, including aldosterone, which affects sodium uptake by the kidney. And many of these signaling cascades are also regulated by even higher level hormones secreted by the hypothalamus and pituitary. So these are just examples of how there's a multi-organ coordination throughout the body is affected through these small molecule mechanisms. So one possibly less familiar metabolic regulation strategy your body uses is compartmentalization. So we can control the way metabolic activity occurs in some ways by keeping them sequestered in different compartments. So one really clear example of this is that we have, by and large, put most biosynthetic pathways into the cytosol and most catabolic pathways inside the mitochondria. That's a good example. So that allows us to prevent fetal cycling and helps us to co-regulate the two different activities independently of one another. The other way we have organized, we as vertebrates have organized these metabolic partitioning is by putting certain metabolic activities into different organs. So for example, the muscles are responsible for heavy exercise. They'll produce lactate. Rather than regenerating lactate into glucose inside the liver, your muscles actually just secrete it directly to the blood. which is why after heavy exercise you get this lactate burn in your legs, as your lactate is being dumped out into the blood, and that's ultimately transported back to the liver. And over a longer period of time, the liver invests the energy into resynthesizing into glucose, transporting it back to the muscle. And this exchange between the muscle and liver we call the Cori cycle. So there's also compartmentalization that occurs within the cell. Like the one example that we talked about before was a difference between reactions occurring in the mitochondria and the cytosol, but there are also a separation of things between the cytosol and the endoplasmic reticulum. One example of that is the breakdown of glucose 6-phosphate into glucose. This is glucose 6-phosphate is both the first committed step in glycolysis, but is also the last step before we secrete it out, if we're going to secrete glucose out into the bloodstream. And so to prevent futile cycling between both the production of glucose 6-phosphate via glycolysis and the release of glucose to secrete it out to the blood, we actually segregate these two enzymes into two different compartments, with the ER holding the phosphatase activity and the cytosol holding the kinase activity. So another mechanism that we use to regulate metabolism, that's sort of new and emerging one that we're beginning to know, is the regulation of metabolism through the formation and disruption of enzyme complexes. A beautiful example of this is the regulation of the pentose phosphate pathway and glycolysis in red blood cells. Now, under oxygenated conditions, the red blood cell needs to produce a lot of NADPH to fight oxidative damage. Under deoxygenated conditions, the red cells need to produce a lot of ATP because it gets squeezed through the capillaries and they actually leak ATP in the process, so they need to synthesize a lot of ATP. So the way that the cells deal with this is that under deoxygenated conditions, where glycolytic demands are low but PPP needs are high, the cells actually form a multi-enzyme complex on the cell membrane on the surface of band 3. And when they do, each of these enzymes, these glycolytic enzymes, binds to band 3, they become deactivated. So by deactivating them, it tends to shuttle flux through the PPP and deactivate glycolysis. Well, as it turns out, deoxygenated hemoglobin will actually bind to that same site on band 3 and displace these enzymes. And when it does, it activates all these glycolytic enzymes and then shuttles flux away from the PPP and into glycolysis. So this is a really nice example of how the regulation of two major pathways can be coordinated through the formation and disruption of this enzyme complex. So these are really beautiful. You can actually see them. You can actually see this metabolic regulation happening in real time. So here's an example of band 3. It's been stained on the outside of the cell membrane. You can see this beautiful biconcave shape of band 3 that's typical of the protein. You can see that it co-occurs with aldolase, the metabolic enzyme. And under these oxygenated conditions, these are all co-localized here to this outside the cell. You can see this for a variety of enzymes. So here's GAPDH, same thing. Under oxygenated conditions, it's all along the cell membrane, same with aldolase, and lactate dehydrogenase, pyruvate kinase. But under deoxygenated conditions, all of these enzymes are distributed throughout the cell, suggesting that the deoxygenation of hemoglobin displaces these complexes and ultimately stimulates their activity. Now, the regulation of glycolysis in red cells isn't the only example of enzyme complexes. There's another example, is the pyrannosome. So the pyrannosome is a multi-enzyme complex. It has 6 enzymes that allow substrate channeling for the production of these purine nucleotides. Now, purines are very energy intensive to synthesize, and it's sort of this long linear pathway, and it doesn't make a lot of sense for these things not to be co-localized. So these enzymes get together, they form channels that pass the substrates from one to another. We'll learn more about the details of this synthesis later. But the formation and disruption of this complex ultimately allow you to regulate the synthesis of the purine nucleotides. So today we discussed a variety of different forms of metabolic regulation. We talked about transcriptional regulation affecting sort of long-term energy balancing, hormones, how they can interact and coordinate multi-organ metabolism activities. We talked about how those hormones can act through covalent modifications. We also talked how allosteric activators can turn on and turn off enzymes. We talked about the role that compartmentalization and multi-organ regulation can play in the control of metabolic flux.